In this video, we will explore NHLBI BioData Catalyst powered by PIC-SURE and provide an overview of the platform, discuss the components of the platform, and cover basic functionality. BioData Catalyst powered by PIC-SURE empowers researchers to access data by facilitating approachable research for all skill levels. Researchers can use PIC-SURE to search the data dictionary at the variable value and genomic variant levels. Once a researcher identifies a variable or variant of interest, they can apply a filter to create a cohort of study participants. Then, after they have refined their cohort and added variables of interest, they can retrieve their selected data frame to kickstart their analysis. This can be done without the need to open any raw data files or map to data dictionaries. PIC-SURE stands for Patient Information Commons Standard Unification of Research Elements. This platform allows users to search, query, and build cohorts at the phenotypic variable and genomic variant level across datasets in NHLBI BioData Catalyst, or BDC. PIC-SURE integrates the clinical and genomic datasets in the ecosystem, which includes studies from the NHLBI's Transomics for Precision Medicine, or TOPMed program, several COVID-19 related studies, and studies from the NHLBI Biologic Specimen and Data Repository, or BioLink. There are two main components to PIC-SURE, the user interface, or UI, and the application programming interface, or API. These components have the same functionality but are accessed and used a bit differently. The UI allows users to conduct searches, explore variables, and build queries using a point-and-click interface. On the other hand, the API allows users to build queries using code and export data directly into an analysis workspace. To access the UI, simply navigate to the BDC Powered by PIC-SURE site. The link to this site will be available in the YouTube video description. The API is accessible through Jupyter Notebooks or R Markdown files, depending on user preference, through either of the analysis platforms on BDC, BDC powered by 7 Bridges or BDC powered by Terra. Users can search for terms in the Google-like search interface and point and click to build queries in the UI. In the API, users utilize Python and R functions to build these queries. Finally, a user can export data from the UI using a dataset ID, which is then used in an analysis workspace to retrieve the data frame. In the API, a user simply runs a query and uses code to export the data directly into the workspace. When you navigate to PIC-SURE, you will be taken to this landing page. Here, you have the option to browse all available data on BDC, or to log in for more functionalities such as exploring participant-level data. Let's review these two options. One option that PIC-SURE provides is to browse all data available in BDC. This is a publicly available tool that allows you to search any clinical variable in PIC-SURE and apply filters to conduct feasibility assessments. There is no dbGaP authorization to access the data required, meaning researchers with no data access can use this to search variables of interest and perform feasibility studies to determine which data to request access to. Because there is no authorization required to use this tool, there are some protective measures that have been implemented. The first is that only obfuscated counts of the participants are returned. There is no access to participant-level data permitted. The obfuscation means that if less than 10 participants meet the inclusion criteria, it will be shown as less than 10. Additionally, only phenotypic destigmatized variables are available for search. NHLBI has defined variables from the following categories as sensitive information that should be excluded: mental health diagnoses, history, or treatment, illicit drug use history, sexually transmitted disease diagnoses, history, or treatment, sexual history, intellectual achievement, ability, or educational attainment, and direct or surrogate identifiers of legal status. The other option that PIC-SURE provides is to explore authorized data, which allows users to access authorized data, apply filters, and retrieve the data for their selected cohort. In order to use this option, you must log in to PIC-SURE. To use this tool, a user must have dbGaP approval to access at least one study. Because the user is authorized to access these datasets, they are able to retrieve both aggregate counts and participant-level data. They can also search and query across phenotypic and genomic variables. Additionally, the PIC-SURE API can be leveraged to explore authorized data, specifically using R or Python coding languages. The PIC-SURE API allows users to query the database using programming languages. Currently, this is available in Python Jupyter Notebooks, R Jupyter Notebooks, or RStudio. Example notebooks with instructions to use the API can be found on the Access to Data Using PIC-SURE GitHub repository. These examples include notebooks to help users get started with basic functionality of the API, code to begin performing genomic queries, examples for specific use cases such as COVID-19 or sickle cell disease, and examples to kickstart harmonization across several studies. The link to the public GitHub repository will be available in the YouTube description.